A very good morning. Today we will be addressing the journal which is titled Frontal Sinuses and Human Evolution. So this was published in the year 2022 on October, and the journal is Scientific Advances and the Impact Factor is 14.14. Terminologies: frontal sinus, we all know the air cavities with frontal lobe located above the orbits, and pneumatization is the process of air-filled cavity formation in cranial bones influenced by genetic, developmental and biochemical factors. Hominins are humans and their extinct relatives post divergence from chimpanzees approximately 6 to 7 million years ago. Endocranial volume, it is nothing but the brain cavity size. It is measured via cranial capacity. And directional asymmetry, DA, it is a consistent left-right differences in morphology. And the spandrel, golden limb, is a non-adaptive evolutionary byproduct of other traits, genetic traits. The relevance of the study is that this study addresses a long-standing gap in the paleoanthropology by systematically analyzing frontal sinus variation across extinct and extant hominids. So novelty is that this is the first large-scale comparative analysis which had 69 fossil hominids of 21 species alongside modern humans and African apes, which include gorilla, and for surpassing prior studies focused on single taxa like Neanderthals and Homo sapiens. Also, this particular study rejects long-standing hypothesis, which is the climate adaptation. So there was no evidence that sinuses evolved from cold adaptation. So this contraindicates Kuhn study, 1962, and Churchill study. This was a very big breakthrough study, and which was published in 1998. Also, it rejected another hypothesis, the masticatory strain hypothesis, which states sinus morphology it does not correlate with chewing mechanism. So this study rejected both, and it introduced the standardized methods like the reproducible protocol for measuring sinus dimensions, both linear and volumetric in both 2D and 3D basis, so which can be used for our previous studies, and it also links sinuses to brain evolution. So the aim of this study was to resolve databases on frontal sinus evolution by testing functional hypothesis and assessing phylogenetic features. The first objective was to compare sinus size or shape across hominids and extinct apes. After which evaluating whether the sinuses reflect climate adaptation or masticatory strain, and to explore correlations between sinus asymmetry and frontal lobe morphology, and to determine if sinus traits clarify hominid evolutionary relationships. So these are the aim and objectives of their study. As said, methodology, they had 69 fossils which included all holotypes of Sahelanthropus, Australopithecus, Paranthropus, and the Homo sapiens species, including Homo erectus, Homo naledi, and Neanderthals. So it also included extant primates, 98 samples of extant primates, which included the pan species as well as the gorilla species, and modern humans, 345 human samples, modern human samples, with geographically diverse population. So CT and micro CT scans, high resolution 3D reconstruction using this Evolutionary software was used, and measurements both linear and volumetric measurements were used, both 2D and 3D. So they measured for the max width, height, and anteroposterior lens. Volumetric, that is for the measuring the endocranial volume. So that was used, and that was compared with cube root transform sinus. And the statistical tests were carried out, and statistical analysis they used the PCA. PCA they differentiated taxa based on absolute or relative sinus dimensions, and manual tests they confirmed no geographic clustering in modern humans was seen, and they also performed regression analysis that examined sinus brain size relationships. Will be discussed below. And the key findings are that the early hominids, Australopithecus and Paranthropus, they resemble the pan species or the gorilla species. Sinuses scale isometrically with brain size, suggesting weak developmental constraints. suggesting very weak developmental constraints. Later, Homo erectus, the now species like Neanderthalis and now current sapiens, Homo sapiens, they have greater variability in sinus size and shape. They have reduced anterior-posterior expansion, possibly due to cranial integration with brain. And this study also revealed that there was more support for adaptivist hypothesis, that is sinus size was unrelated to climate, which was contraindicated contraindicated compared to the previous studies. And it also said there is no correlation with masticatory biomechanics, which was Churchill's study. And brain sinus covariation, regarding that, left sinus enlargement, it correlates with the right frontal metalia. It was consistent from Homo erectus onwards, Homo erectus and Homo sapiens. It is evident, and taxonomic insights reveal that it supported distinct groups like Homo rhodensiensis, and this was Broken Hill study, which is Homo heidelbergensis, and Homo naledi clusters with Homo erectus, reinforcing it is all placed in the Homo species itself. So these were the major key findings, and evolutionary implications. So the sinus morphology, it aligns with clinical traits aiding species delineation. That is the middle Pleistocene hominids, and developmental integration was also seen. It suggested sinuses are influenced by brain growth patterns, not just bone structure. So this study, it emphasized that there is developmental integration. The strengths of the study, it is standardized, and it is well standardized, and bilateral analysis was done in this study. This accounts for the asymmetry. Since the both frontal sinuses, they are not symmetrical, it is asymmetrical, and plus this was crucial for brain sinus link. And it had a comprehensive data set of frontal sinuses from the Australopithecus species till date. And it combined 3D imaging systems with multivariate statistics, which was innovative, and it integrated paleoanthropology, anatomy, and evolutionary biology. This is the largest fossil data set for frontal sinuses till date, and it combined imaging, morphometrics, and evolutionary biology all together using this 3D, and it also used very rigorous statistical analysis to robustly test this hypothesis. And this study also has open-ended conclusions, so there is no over-interpretation. And this study, it highlights the need for developmental studies. The limitation is it has geographical gaps. There is limited representation of some hominid population, because since it is all retrieved from a very earlier database, we were not able to find hominid populations. And authors, they acknowledge the incomplete fossils, but maximize data from available specimens are required. And extent comparisons, more gorilla pan species were very less compared to the Homo species. And fossil preservation, fragmentation limits bilateral comparison. That is, for example, if there is missing right or left sinuses, and as said, that where some samples had less data with regard to taxa. The fossil represent the key taxa were missing. For example, like early Homo species habilis, the data was missing, and fragmentation, it limited bilateral analysis. Developmental data was also lacking, since we had no data to track sinus growth versus brain development, and functional tests were indirect. Biomechanical modeling, example FEA, it could further test mastigation hypothesis, because this study said that the mastigatory strain was not relevant to the available literature. Future direction, we can expand fossil sampling by including more H. florensis, decinovans, and early Homo species in the study. And we can also compare sinus growth with brain endoplasm juveniles and explore genetic base of sinus variation, for example, RUNX mutation. And we can compare primate models, add Congo and hyoblades to assess ancestral states prior to the Homo species and the Australopithecus species. So to conclude, this study redefines frontal sinuses as a phylogenetically informative trait rather than a biomechanical adaptation. So this study provided a very robust framework for analyzing internal cranial anatomy in hominin evolution through fossil fragment. Though fossil fragmentation it remains a challenge, these findings emphasize the importance of integrative approaches in paleoanthropology. So the bottom line is this study is a strong contribution. Though there is a very limited fossil record bias, this study shifts the paradigm on frontal sinus function, emphasizing evolutionary relationship over adaptivist explanation. Future work should prioritize developmental biology and expand fossil sampling. So this study, it was assessed based on the tripod transparent reporting and fossil guidelines. This fossil guidelines, it is for paleontological based studies. So the study design, the study design was good, and the priori hypothesis that was the climate and the mastication strain theory was rejected, but brain sinus link was exploratory only. So it can be, it was not what it is partial. It can be, validated. Sample selection was good, but the modern human population was diverse. It lacked plant species and gorilla species. So that sample could have been included. And the data collection, the standardizing image protocols were formed, but blinding was a single observer study. So that was actually a potential bias. And endocranial volume was used, but the facial prognostic values were not added. So that could have been a parameter. And statistical methods, geographical comparisons were not made. It was all diverse. It was diverse, but still geographical comparisons were not made. And it only used raw data was accessible, but still the effect sizes were partially reported. So R value correlations were done, but Cohen's test was not done. And interpretation was good, reproducibility. The data was in paper, no DOI was there. There was no DOI. And so this is collected from the archival collections. So